Hello, and welcome to this brief introduction to bioinformatics. I'm Robert Morell. I'm the director of the Genomics and Computational Biology Core. The GCBC is a shared core facility between the NIDCD and the NIDCR. So I am the NIDCD, NIDCR, GCBC director. Many of you are new to the federal government, but you've probably already realized that there's a bewildering amount of acronyms that you have to learn just to read your daily email. That's certainly true of bioinformatics as well. But that's one of the goals of this brief introduction, is to introduce you to some of the terminology and the acronyms.
The goals for this talk are to introduce the Genomics and Computational Biology Core, to give a brief description of how we generate the data, to survey some common bioinformatics terms and their acronyms, suggest ways to look at your data and generate testable hypotheses from it, and also to introduce available resources for analyses and training. This slide presentation will be available to you in print form, so you will have a copy of all the web addresses, all the email addresses, etc. There's no need to frantically copy everything down as we go. The Genomics and Computational Biology Core is a shared core facility between the NIDCD and the NIDCR. Shown here is our somewhat out-of-date website, but the best way to contact us is by email, and my email address is here.
So what does the GCBC do? We help to design your genomics experiments. You supply the RNA or DNA, and we'll analyze it for quality and abundance. We'll make sequencing libraries, and then sequence them on either a NextSeq 2000, a MiSeq, PacBio Sequel II, or an Oxford Nanopore MinION. We'll analyze that sequencing data, and we'll document library production, sequencing, and bioinformatics methods for you. In other words, all those bits and pieces that need to go into your methods section. We'll generate figures and tables for publication, and finally, we'll help upload the data to a public repository. In the last sane year of Western civilization, that is fiscal year 2019, the GCBC worked on 72 projects and made well over 1,000 libraries. We handle a variety of projects, but the overwhelming majority are RNA-Seq. Consequently, I'll focus on RNA-Seq in the next few slides.
The first step in an RNA-Seq project is to identify which RNA you want to characterize. Most studies focus on polyadenylated RNA found in the cytoplasm, which includes all of the protein coding sequences. Poly-mRNA comprises only about 1 to 3% of the total RNA in the cell, so it's usually enriched in subsequent steps from total RNA. But realize there are methods for characterizing the other RNA species in a cell. For most RNA-Seq applications, we request that the investigator provide 250 nanograms of total high-quality RNA. RNA quality is measured in a RIN, or RNA integrity score, which is a scale from 1 to 10. RIN scores above 7 are usually considered good quality. We can advise you on how to recover RNA with RIN scores of 10. How much RNA is 250 nanograms? Well, the typical cell has about 10 picograms of RNA, so 25,000 cells would be required to generate 250 nanograms. But these are ideal numbers, and they're the numbers that are typically required by a commercial vendor. We have made many successful libraries for much less and much worse material than this. The most important number on this slide might be 4, because for any statistical analysis comparing 2 or more groups, we recommend a minimum of 4 biological replicates for each group, and I'll explain why later in this talk. Whether starting from genomic DNA or RNA converted to cDNA, the next step in the process is to fragment the DNA into approximately 300 base pair pieces, and then add adapters to the ends. There are a variety of ways to accomplish this. I'm showing one method, which relies on a transposase to both fragment and add the adapter. The end result is a library in which the DNA fragments have ends that are complementary to capture oligos, which are seated in lanes on a glass slide. Single-stranded library fragments are flowed onto the slide at low concentration, and the fragments are captured. The fragments are surrounded by thousands of unbound capture oligos, and the unbound end of the fragment will find its complement, forming a bridge. The second oligo serves as a primer to replicate the second strand. This continues for several cycles of PCR bridge amplification until there are colonies that are thousands of copies of the original captured fragment. The formation of the bridge for amplification places an upper limit of about 800 base pairs on the size of the fragments that can be efficiently rendered into PCR colonies. The PCR colonies, or clusters, are then subjected to sequencing by synthesis. That is, single-stranded templates are replicated using fluorescently labeled nucleotides. The addition of each fluorescent nucleotide is imaged by camera in photographic tiles that span thousands of clusters at a time, each cluster being thousands of replicates of an original library fragment. This is why the inherently ambiguous term next-gen sequencing is better replaced with the more descriptive term massively parallel sequencing, which was the original term used for this technology. Note that a further size restriction is imposed at this point. The 200 to 800 base pair fragments are only sequenced 35 to 150 base pairs in from each end. The output data is a series of images recording the color changes at each cluster, which is decoded as a DNA sequence in a FASTQ file. I will describe FASTQ files a little later in the talk. Almost all of the genomic and transcriptomic data generated in the last 20 years has been from this kind of short read format. This is ideal for identifying local features in reads aligned to a reference genome, such as sequence variants or ChIP-seq peaks. It's also useful for counting the number of transcripts in a cell, if you only require that the read match somewhere in the transcript. But short reads have limitations in our ability to resolve, for example, the exon usage of mRNA isoforms, which average over 2,500 base pairs, or resolving large genomic variation, especially copy number variants. That is why the GCBC is transitioning to long read sequencing, specifically to PacBio sequencing, although we are finding uses for the noisier nanopore sequencing platform. Here is a comparison of the sequencing platforms currently in use in the GCBC. Our two Illumina short read sequencers, the MySeq and the NextSeq 2000, generate millions of high-quality reads. The raw error rate for these platforms is 0.1%. That is, 1 base in 1,000 is an error. The Oxford Nanopore MinION and the PacBio Sequel II are both capable of sequencing greater than 100,000 base pairs a continuous sequence, but at much lower throughput and much higher error rate. Most importantly, the Sequel II is also capable of sequencing fragments up to 15 kb with extremely high accuracy. PacBio sequencing is also essentially sequencing by synthesis, with the imaging of single additions of fluorescently labeled nucleotides. Here, however, the image is a continuous movie of the additions to a single growing strand of DNA. This is single molecule sequencing. The high accuracy is achieved by circularizing the DNA template and then sequencing the circularized template over and over again in a continuous loop, and finally building a highly accurate consensus sequence from the several passes. The circularization step imposes a size limit of up to 15 kb, but that's ideal for sequencing cDNA, for example. Also, because of technical details beyond the scope of this talk, the accuracy can be several orders of magnitude greater than the best consensus sequence achieved with Illumina short read sequencing. The limitation again is the throughput. So PacBio sequencing is ideal for discovery of isoforms or genomic variation in small samples, but it's not presently suitable for quantitative methods.
So now you have an idea of how the data is generated. Let's survey some of the terms used for the output files and downstream analysis.
The output from an Illumina or a PacBio sequencer is ultimately a FASTQ file. That is a FASTA file, which stands for fast alignment, which is just the sequence with some metadata in a header line, plus the quality, the Q part, which are quality scores for each base determination. These files are typically several gigabytes in size, since a typical RNA-Seq library will have more than 30 million reads. They can be opened in a text editor, but they're really too large to be efficiently handled in that way. FASTQ files are usually what are required for depositing genomic data in a public repository. In any case, the raw sequence is not useful unless annotated in some way. This is usually achieved by aligning it to a reference genome along with annotation tracks describing the location of specific features in the genome. The location of the gene coding regions are provided in a GFF or GTF file. GFF stands for General Feature Format. Most vendors will use RefSeq as their annotation track. The GCBC uses the GenCode annotation since it has almost twice as many annotated genes. For differential gene analyses, that is important since your read can only be annotated by what is in the reference GFF. It's also important to keep in mind when you are comparing your results to other studies in the literature or in the public data repositories. Always check the reference genome and annotation. Are you using the same reference genome version and the same annotation? The Venn diagram illustrates that the three main annotation resources, GenCode, RefSeq, and the UCSC, largely overlap in their transcript annotations, but there are significant differences. BAM files are binary versions of SAM files. That is, they are compressed and not readable in a text editor. If the BAM file is the output of an alignment to a genome, the header information will include details on the alignment and the quality scores will relate to the alignment. This one is showing that the match is to chromosome 1 and to two 46 base pair regions separated by a gap of 648 base pairs. This is likely a read from a cDNA, matching two consecutive exons separated by an intron. There's other information here that we need not discuss now. Some public repositories prefer BAM files to FASTQ files. BAM files can be manipulated with the SAM Tools suite of programs, and you can view BAM files on your desktop computer using IGV, or Integrative Genome Viewer. In IGV, you'll see the reads aligned to a reference genome with a variety of annotation tracks that you can choose, in this case the RefSeq GFF. Here we are looking at reads aligned to the gene grainy head-like 2, with colors indicating mismatches that might be sequencing errors or they might be natural variation. The top coverage track shows a histogram of the number of alignments in a particular region. The junctions track shows a histogram of reads that span exon junctions. There is a wealth of data output from an RNA-Seq study. I'll review a typical RNA-Seq output and suggest how to review the information. First, there are always multiple figures which summarize the results. I suggest you start with the PCA plots, or Principal Component Analysis plots. There is a wealth of variation between the large data sets for each of your samples. Some of the variation is technical, introduced during sample handling or library construction. Some is stochastic or random, and some is individual variation irrelevant to the hypothesis you're testing. But hopefully, some is biological variation relevant to the hypothesis. A PC analysis attempts to parse the variation into recognizable components. One can then try to correlate known attributes about the libraries to those major components. In this example, comparing reads from wild-type and knockout utricles, it is apparent that one of the wild-type samples, number 4, deviates from all the other samples, knockouts included. There are statistical tests to determine whether a sample is a true outlier, and wild-type number 4 was determined to be a statistical outlier in several purely technical aspects. We can remove wild-type number 4 without compromising the analysis because we still have 3 biological replicates, the minimum number for a statistical analysis in the wild-type group. Reanalyzing the data after the removal of sample wild-type 4 shows that the samples are now well separated by genotype. And comparing the DE gene lists before and after the removal of sample wild-type 4 demonstrates that the results can be dramatically different. Nearly 2,000 DE genes were revealed after the removal of this technical noise. When planning their RNA-Seq experiments, investigators often ask whether it is more important to have more samples or to sequence the samples to a greater depth. The answer is that the number of DE genes detected is strongly correlated to the number of biological replicates in the study, while the total number of reads has little effect on the ability to distinguish true versus false positives, since the ROC curves quickly reach saturation. So, more replicates are better than more reads. Also, for gene counting purposes, it hardly matters how long the read is. In a recent study, we compared the DE results obtained from the original 150 by 150 paired end reads to that obtained by trimming all the reads down to 37 by 37. As you can see, the red and green circles representing the results are almost congruent. There's less than 1% difference. The GCBC standard output also includes the scripts executed in the analyses, so you or anyone else can reproduce the analyses. This includes the scripts used to generate all the plots and figures, so you can modify them if you choose. And I'll have more to say on that a little later. All of the FASTQ and BAM files, as well as quality metric reports and other tables and reports generated during the analyses, are included. Some of these may be required to be deposited in a public repository in order to comply with journal requirements and the NIH Genome Data Sharing Policy. An RNA-Seq analysis will generate a list of several hundred to several thousand differentially expressed genes. The question is, what to do with such a list? There are many commercially available softwares for analyzing the gene lists, some of which you can have access to through the NIH library. But we recommend 2 freely available softwares, starting with Enricher, which is maintained at the Mount Sinai Medical School in New York. Starting with a list of genes, Enricher will search public databases and return annotations that are enriched in your gene lists. These include metabolic pathways, transcription factor associated genes, disease pathways, developmental pathways, particular cell and tissue types, known drug response pathways, a large list. The initial lists are basically just lookup routines, but there are other features that allow you to explore associations computed from your results that might constitute relationships beyond what is annotated. Another recommended web page is Top Cluster, maintained by Cincinnati Children's Hospital Medical Center. Again, starting with a gene list, you can compare gene lists from multiple RNA-Seq studies or specific tissues to identify common or unique pathways correlated with those lists. This can lead to a rich set of annotations to help understand the biology represented in the DE gene lists. Here are some suggestions on how to generate the input DE gene lists for these or any downstream pathway analyses. Shown here is the output of the DE analyses which we report in an Excel spreadsheet. We run 3 software packages for our DE analyses: DE-Seq2, EdgeR, and Limavoom. These softwares differ mainly in the way that the gene expression data is normalized. There's no single right way to do that. Each software has its proponents, but we choose to run all three and let you compare the results. Generally, Limavoom is the most conservative and will generate the smallest DE list, while DE-Seq2 typically generates the largest. But it's important to note that the lists show considerable non-overlap. In other words, the smaller Limavoom list is not necessarily just a subset of the larger DE-Seq2 list. The lists are slightly different, again because of the different normalization methods. So you may choose one list over another, or you may choose to make a list that's the union of the three results, or maybe just the intersection of the three, or you may want to do both and compare. Next, notice that both the p-value and the adjusted p-value, or the FDR in the case of EARR, is reported. Use only the adjusted p-value, or the FDR. Ignore the p-value, no matter how enticing it is to see a phenomenally small p-value next to your favorite gene. Remember that each gene list is tested for significance, so you are looking at the results of perhaps 40,000 or more statistical tests. You must consider only the p-values corrected for so many tests. And finally, it's best to pay attention to the direction of expression change in your comparison. In this example, the comparison was between samples from a fibrous dysplasia biopsy and normal tissue. In DE-Seq2 and Limavoom, the log2 fold change will be positive when the gene is relatively upregulated in the FD tissue, and negative when the gene is downregulated in FD tissue. So you may want to generate separate gene lists for those that are upregulated and those that are downregulated. Single-cell genomics is extremely popular. The GCBC uses the 10x genomic system, in which individual cells from a single cell suspension are captured in oil droplets along with a bead, which is coated with capture oligos, in this case poly-T oligos to capture mRNA once the cell is lysed. The reverse transcriptase reaction occurs in the lysate, in the presence of many inhibitors, so for each cell the reaction is inefficient. A barcode unique to each bead is incorporated into the resulting library, so when all of the reads are sequenced, they can be assigned back to a cell of origin. The barcoded library pools are computationally clustered together based on the global similarities of expression. The shallow sequencing from many similar cells can, in the aggregate, reconstruct the transcriptome of a cell type. The data is analyzed initially with Cell Ranger software, and the output is very different from that of tissue-level RNA-Seq. To view this output, you'll need 10x's Loop browser. In Loop, you can browse the clusters and search for specific genes, but realize that single-cell RNA-Seq is very shallow and very noisy, and your gene of interest may not be represented at all. Also, the clusters generated by Cell Ranger are not necessarily the best that can be achieved, and there are mathematical constructs that may or may not correspond to biological cell types. In order to make the most of your data, you will need to analyze it further with Surat. You will be able to annotate the clusters, but more importantly, you'll be able to bring the biological information you know about the cell types represented in your data set to influence the algorithms for determining clusters. Analysis of this kind is highly iterative. The more you learn about the data, the more information you can provide the software, which in turn adds to the resolution of the cell type clusters. That is where you bring your biological knowledge to bear on the analyses. The web page for the Satija Lab has many excellent tutorials in using Surat, but in order to use Surat, you need to learn R. Before discussing R, I should quickly mention Spatial Transcriptomics, or Visium, because I'm often asked about using this 10x genomics technology in the inner ear. Here, instead of capturing the RNA with oligos coating a bead, the capture oligos are arrayed on a glass slide in dots that are 55 microns across. Each dot is comprised of thousands of barcoded oligos. When a tissue section is laid on the glass slide, the RNA will leach from the tissue and be captured by the oligos lying immediately underneath. The captured RNA is reverse transcribed and the resulting libraries are sequenced. The reads can then be assigned to a dot of origin, and thus to a discrete region of the tissue section. The GCBC has worked on spatial transcriptomics projects, but not in the inner ear. The reason is that the spatial resolution needed to be practically useful in characterizing the highly patterned organ of Corti is much greater than the 55 microns now available. Everything within a dot with a 55 micron diameter will appear the same in gene expression, and as you can imagine from this illustration, that would hardly be informative in distinguishing the transcriptomes of hair cells from supporting cells, for example. However, 10x Genomics has announced that it will achieve a 1,000-fold increase in resolution in the near future, so this may become an area of active research in the inner ear soon. So, how to get started in acquiring some of the skills you need for bioinformatics? As I said a moment ago, in order to analyze single-cell data especially, you will need to learn R. The good news is that R is relatively easy to learn, and learning R is worthwhile well beyond single-cell analyses. Virtually every current bioinformatics pipeline is represented in R, and there are many R packages that extend data analyses far beyond bioinformatics. You can download R from the CRAN repository website listed here on the screen. It can run on any workstation. I recommend that you run R in an environment called RStudio. In RStudio, you can simultaneously see the command line environment you are executing and also see, for example, the graphics you are generating or the data objects you are creating. Most R packages are available through Bioconductor, which is also a good place to find tutorials to learn R. I mentioned before that the standard GCBC output includes the scripts to run the analyses and generate the plots and graphics. Shown here is the DE-Seq2 R script. You can load this into RStudio if you wanted to reanalyze the data or to modify any of the plots. The GCBC also maintains a server we call Bricks, which has pre-installed R packages for use by intramural researchers. There are also short video tutorials on how to use some of the applications, and this is ideal for people who need only to occasionally customize a heat plot or a PCA plot for publication. But the best option is to get an account on the NIH high-performance computer cluster called Biowulf. This is one of the largest compute clusters in the world. On Biowulf, you can run analyses much too large to run on your workstation. All of the softwares listed in this presentation are installed on Biowulf, including R and RStudio. Sometimes, in order to compare analysis results with published results, you'll need to analyze the data with an older version of the software, and that's another advantage of Biowulf. You have access to not only the current version of a software, but also older versions going back several years. The operating system for Biowulf is Linux, so you'll need to learn some basic command line computing. On Windows, request IT to install PuTTY in order to log into Biowulf. If you have a Mac, you already have an SSH client installed because the Mac operating system is essentially Linux. On the Biowulf page, you'll also find information on how to run graphical programs like RStudio or IGV. You can access the data from published RNA-Seq studies through the Genome Expression Omnibus, or GEO. In fact, the data you generate will likely need to be uploaded to GEO in order to comply with journal requirements. But for specifically inner ear expression data, the best online resource by far is GEAR. There was an excellent tutorial on GEAR earlier in EARssentials, so I won't belabor the point, but I'll just remind you that there are excellent tutorials to help you get started, and I encourage you to log on and create an account soon. Information on the genes that cause hearing loss is available at the Hereditary Hearing Loss webpage and the Deafness Variation Database, shown here. You can also get a good introduction to inner ear genes, anatomy, and physiology through these web pages. There are plenty of resources to get training in bioinformatics, including the Foundation for Advanced Education in the Sciences, or FAES. FAES hosts workshops, short courses, and hands-on multi-day classes on a variety of topics, including bioinformatics. And I already mentioned that you can find training through the Biowulf webpage. The NIH library has regular classes, especially in those bioinformatics programs that you can access through their site licenses. The Center for Information Technology, or CIT, also has regular classes in computing and bioinformatics. Finally, another good way to learn about courses, seminars, workshops, etc. is to join a bioinformatics-related scientific interest group and to subscribe to their listserv. So that's it, or at least that's as much as I can cover in this amount of time. I'll entertain any questions you have either now or feel free to contact me via email.